Science on Surfaces.
Hello there, and welcome to this podcast, Science on Surfaces, Tips, Tricks and Tools, with me, Malin Edvardsson. So today we will talk about a really popular topic. We will talk about the microbiome and probiotics, and we will be talking about recent research related to the interaction between probiotics and milk fat, and how this may affect bacterial adhesion in the gut. So here with me in this call, I have Dr. Joana Ortega-Anaya, postdoc researcher at the Department of Food Science and Technology at The Ohio State University. And Joana specializes in the study of milk components and how they affect probiotics and human health. So welcome, Joana.
Hi, thank you for having me, Malin.
Yes, great to have you here. So how are you today?
I'm very good.
Great. So, like I said, the topic of the microbiome and probiotics, it's become so popular recently. There's much discussion on the importance of the microbiome for our well-being. I read somewhere that it's even called the supporting organ because it plays so many key roles in the daily operations of the human body. So, if we just start with the basics, so what is the microbiome?
Yeah, you're right, and it's a very interesting topic. So the microbiome is referred to as a group of microorganisms that are in a certain environment. So it could be a small environment like an organ or a small animal or a plant. But it's a very specific and characteristic collection of microbes. Sorry. So in humans, we have a few microbiomes that are very important. One of them is the skin microbiome, or the skin collection of microbes. We have the oral microbiome, which is the mouth. We have the respiratory microbiome, which is the nose, the lungs, and all that system. We also have the urogenital microbiome, and the most important, or the one that has been receiving a lot of attention, is the gastrointestinal microbiome, or also called the gut microbiome. Maybe you have heard it like that, the gut microbiome.
Yes.
And it is very important because so it spans from the stomach, then the intestines, the small intestine, and then the large intestine. And it's very important because the intestines are connected to the bloodstream, right? So what happens in the gut has a direct impact in our health in the end, because through the bloodstream, whatever happens in the gut gets transported through to blood vessels and organs.
Okay.
So that's why it's so important. There's a connection between what happens in the gut and what happens in our organs. From now on, I'm gonna be talking about the gut microbiome, because this is what we are interested on.
Yeah.
The gut microbiome, you may or may not know this, but it's largely affected by many factors. It's a very complex environment, and it's not a one-way street. Okay, it's a two-way street. But in general, it gets affected by where we were born, where we live, like the geological location of our bodies. Of course, the diet. If we have, if you're feeling sick, if we have a disease.
So how does this work? There are so many factors that you mentioned
I
now.
know. Yeah, exactly. Well, nobody really knows. That's why it's so complex, because also your genetics play a role in your microbiome. The way you were born, if your mom had a vaginal birth, or if it was through a C-section, that affects completely your microbiome. So there's many factors that affect your microbiome. But the point is, when you get sick, or when there's something affecting your, let's say, you're stressed, and that can modify or change the equilibrium of the microbes that are in your gut, because you have a certain amount of pathogens in your gut and a certain amount of non-pathogens or beneficial bacteria in your gut. There's always that population, and they have a specific equilibrium. So when something happens and that equilibrium gets modified, it might be that some pathogens proliferate and the non-pathogenic bacteria are not proliferating as they should. So that equilibrium gets modified, and then you can have specific alterations in your health that can go from, just feeling a little sick in your tummy to having colitis or infection, diarrhea, and then, from then on, it can be even cancer, right? But like I said, it's very complex. There's not one little point or one little thing that you can say, okay, if I do this, I'm not going to get this disease. It's very complex because bacteria is very complex. That equilibrium in the gut is very complex, and we as humans are very complex as well.
Yeah, no, I was thinking, you mentioned we have like the good microorganism, or like the ones supporting us, and the ones that are, not really supporting us, and we affect that equilibrium or the balance with our lifestyles. So then I guess we can do less of the things that promote the bad guys and more of the things that promote the good ones.
Exactly. So it's a good diet, exercising, because believe it or not, if you don't exercise, that affects your microbiome. And there's also something really interesting called the gut-brain axis. Have you heard of it?
Yeah, I've heard, but please explain more if you can.
Well, it's just the connection that there is between your gut and your the health of your brain. So, and it is largely dependent on the microbiome, believe it or not.
Okay.
So if you're balanced, it's on perfect conditions, it's in optimum conditions, then they say that your brain, you know, could be better, in a better state. And otherwise, if you get stressed, if you have a certain mental condition or a mental disease, that also affects the microbiome and vice versa,
Okay,
So it's not a one-way thing.
Okay, it goes both ways.
Yeah, exactly.
Wow.
So there's a lot of research being done these days, and nobody really understands how it starts, and what's the starting point, how to end it, how to change it, how to really regulate it. But it's been done.
So we can promote the good bugs with some lifestyle factors. You mentioned diet. For example, I know that we can eat probiotics, which relates to your work a bit. So what's with the probiotics? I mean, why is that good for us?
Exactly. So when we have that unbalance in our microbiome, we can do something about it. Like you said, we can ingest good bacteria that can help us change that balance into the optimal state that we need. And for that, we use certain bacteria that are called probiotics. Actually, it could be bacteria or yeast so far. And that bacteria, I'm going to refer from now on to bacteria, probiotics. The World Health Organization defines probiotics as live microorganisms that, when ingested, provide a beneficial effect to the host. Okay. So, thinking of in general, we can have probiotics for the skin, for certain different animals, for dogs, for cats, and of course for humans and for the human gut. The probiotic label, it's dependent on the strain. Okay, so if you have a group of bacteria, let's say Lactobacillus, not all Lactobacillus can be probiotics. Okay, and not all probiotics are Lactobacillus. We have different types of probiotics, like bifidobacterium, enterococcus, streptococcus. But the strain, the specific strain or the species of that bacteria is what's gonna be different. So one strain of the same lactobacillus can be probiotic and the other can't.
Okay, so do we know which strains are working and which aren't?
That's a whole other world.
Okay.
There is a list that gets published by the World Health Organization regarding the bacteria that are considered probiotics up to this date, but that list changes.
Mm-hmm.
Why? Because the mechanism that the bacteria use to be probiotics is different in each and every one of the strains. So it's a very complex, it's a very complex subject. It could be because it generates maybe bacteriocins, which are molecules that prevent pathogens from growing. It could be because they secrete other components, other proteins that help the gut barrier function, or that those cells in the intestine that line the intestines, or it can be something different.
Mm-hmm.
And that is not currently and perfectly characterized to these days. But one thing that's interesting is that for a probiotic to be considered probiotic, the first thing that it needs to do is to endure the gastric conditions, you know, the low pH, the bile salts, and all that. And once it reaches the duodenum, the first part of the intestine, what needs to do at that point is to adhere.
Okay.
The adherence is pivotal for a bacteria to be considered probiotic.
Okay.
And why is that? Because all the microbiome is on the intestinal mucosa. That's where everything happens, the intestinal mucosa. So all of those bacteria are adhere in that intestinal mucosa. So if you're going to ingest a probiotic, the first thing that it needs to be done, it needs to do once it enters the intestine, is adhere to intestinal mucosa.
And how can that be tested? I mean, how do we know that it will adhere?
Well, currently, actually, there are many biochemical tests that you can do, like hydrophobicity, and you can even go to cell culture and measure exactly how they if they bind, and how they bind, how many bacteria bind to the intestinal mucosa. But cell culture is always a very expensive methodology. It takes a lot of money, and of course, not everyone has a cell culture facility in their labs. So they either go to biochemical tests that are not really reliable, or they don't mimic what's happening really in the intestines. So my idea was to, first of all, test that adherence using quartz crystal microbalance, but also going back to the probiotic phenomenon. The more adherence a probiotic has, the higher the residence time is in the mucosa, and it will be able to exert their probiotic effect by whichever mechanism it may do. So my idea was to test the 4 strains of probiotic bacteria, Lactobacillus, using QCM, and also try to make it a better probiotic. Okay, that was my main goal. Try to make it better. But how do you make it better? Well, I work with dairy science, dairy chemistry, and everything dairy. And we work with something called the milk fat globule membrane.
Okay, and what is that?
The milk fat globule membrane is something that's found in all the mammalian milk that there are. So human, bovine, goat milk, any kind of milk. And the milk fat globule membrane is a trilayer membrane composed of phospholipids and proteins, membrane-bound proteins, and these surround each and every one of the fat globules during lactation. So this is formed by the mammary gland cells during lactation. And then once the milk is secreted, each and every one of the fat globules is surrounded by this milk fat globule. But the thing is, we don't know a lot of the milk fat globule membrane and the impact in human health. We do know some stuff, but we don't know exactly how things happen. But like I mentioned, there's two groups of molecules that form the milk fat globule membrane, which are the phospholipids and the membrane. And I was interested in using phospholipids. Why? Because they have been proven to modify the microbiome of pup rats.
Okay.
And modify it so they are more consistent with the microbiome given by breastfed rats.
So would that be just so the lipids themselves, or the lipids
The
in
lipids
some?
themselves,
Okay.
exactly. And they have been shown to have a positive effect in the cells in the intestines, preventing inflammation and just enhancing the intestinal environment. But those experiments usually, if they're not done on animal models, they're done on cell culture using KQ2 cells, which are intestinal cells, epithelial cells, but they don't produce mucosa.
Okay.
Okay, so all of those experiments, they need that extra step where you will be able to see if the bacteria bind to the mucosa. So for this paper that or this research that I have been doing, I used a co-culture, which is comprised by KQ2 cells, epithelial intestinal cells, and goblet cells. Goblet cells are naturally found in our intestines, and those produce mucosa.
Okay.
So we would have a much a better intestinal model.
Yeah. So it's more similar to what you would find. Yeah.
So what I wanted to do was to take phospholipids from the milk fat globule membrane and give it to the probiotic bacteria or the Lactobacillus bacteria, make them adapt to having those phospholipids while they grow.
But so these, I mean, so the cells, the probiotic bacteria, they are basically then surrounded by components that you would find in milk. Is that what you're saying?
No. I actually did a simpler medium.
Okay.
To make the bacteria use those milk phospholipids.
Yeah.
And so see what happens to the bacteria.
Okay, but these are components that you would find in milk that you will buy in a store.
The milk fat globule membrane?
Yeah.
No. The milk fat globule membrane, you can find them on milk, but they're on very low concentration.
Okay, I see.
Currently, there's not an ingredient. There are a few commercial ingredients that are enriched in the milk fat globule membrane, but they are used for infant nutrition.
Oh, okay, I see.
There's nothing you can go like you cannot go to the store and buy milk fat globule.
So it wouldn't be a component of like if you yogurt or something like that. It's a different product that you would have to.
It's different products. In yogurt, you can find probiotics.
Yeah.
Okay, but you don't find them with the milk fat globule membrane.
Okay. Now I was thinking that maybe if they were both in the same product, that would be beneficial. No.
That's the point. By doing research on the milk fat globule membrane, what we want is for the industry, for everyone to start looking at the milk fat globule membrane components and start using them in commercially available products. But of course, we need more research because there's not enough yet to start putting everything together, right? Because like I mentioned, the relationship between bacteria and phospholipids and then humans, it could be tricky. Not everything is known.
No. So if we go back to your research, you have this model system. And so what did you see?
We know that we have 4 strains of Lactobacillus.
Yeah.
And then I'm feeding them milk phospholipids, right?
Yeah.
Once I fed them, I want to study what happens to them. Okay, everything that, because there are not a lot of reports regarding that. So the first thing I did was to look at the membrane morphology of the bacteria. Okay, what happened? Did I see any changes in the bacteria by taking those milk phospholipids? Because sometimes you can give something to a bacteria and they will completely burst or die, or something else. So I wanted to see what happened. And what I saw was 2 of the strains that I used, Lactobacillus acidophilus, and hold on, let me just double check my images. I used Lactobacillus acidophilus, Lactobacillus casei, Lactobacillus delbrecki, and Lactobacillus plantarum.
And these are found in commercial products.
In commercial products, and also, commercial products, not necessarily dairy. For instance, Lactobacillus plantarum, you can find it on fermented meat or fermented vegetables like sauerkraut and stuff like that.
Yeah, because I recognize the names, so I think I've seen them.
Yeah, you can find them there. So that's why I wanted to study these strains, because they are commercially available or available for the diet. Okay. So what I saw that two things happened via looking at that membrane or at that cell by transmission electron microscopy. And what I saw was that 2 of the strains accumulated the milk phospholipids on their surface, like it was kind of added, taken up by the membrane of the bacteria, and I was able to see the little clusters of phospholipids, right?
Okay.
And that was very interesting because nobody really has seen that before. So out of those, Lactobacillus delbrecki and plantarum were the ones that took up those phospholipids on the surface. The other 2 strains, Lactobacillus acidophilus and casei, I didn't see anything different from the controls, which were the cells that were not fed those milk phospholipids. I didn't see anything in those images. And I thought, okay, is this related to adherence, to adhesion? How does this, relate to all of that, which is, what I wanted to study. And what I did next was to see the setup potential of the bacteria. The setup potential is largely affected by the environment and, of course, the biochemistry of the bacteria. So I thought, if I see something different in the setup potential of the bacteria, I might see something. I can relate it to the accumulation of phospholipids in the surface, and maybe I can relate it to the binding, okay, or the adherence of the bacteria. And what I saw was that only 3 of the strains decrease the setup potential value or the electrostatic charge of the bacteria. All of them decreased, and just one of them, Lactobacillus plantarum, was the one that I didn't see any change in the setup potential, but I did see an accumulation of the milk phospholipids in the surface. So I thought, well, what does this mean? And so at this point, what I wanted to do next was to use the QCM and see how everything kind of relates to adherence in a gold sensor. And the reason I used a gold sensor is because I was able to see if the interaction or the adhesion was mediated by electrostatic interactions, right? So that's why I used the gold sensor, and I found something really, really interesting. And what I found that all of the adherence or adhesion or interaction with the gold sensor was modified by the treatment. So the milk phospholipids did affect the binding of all the strains. So they're doing something. They're definitely doing something. In the case of Lactobacillus acidophilus case and case, it increased the adherence to the gold.
So this is the strains where you didn't have any uptake on the membrane.
Exactly.
But still you see
Exactly.
Okay.
So what this means was that even though I wasn't seeing phospholipids in the surface, they probably took the phospholipids and metabolized them somehow. And that affected the binding or the difference in the adhesion. That's how I was able to accumulate the difference in the adhesion. Whereas the other strains that did accumulate milk phospholipids in the surface, for Lactobacillus delbrecki, I did see a decrease in the adherence or the adhesion. And for Lactobacillus plantarum, I didn't see any difference in the amount in the binding or the amount or the mass added to the gold cell.
So 2 increased adherence, 1 decreased, and 1 was no change of the strains.
Exactly. And I really liked using the QCM because I was able to do kinetics, from the frequency. I was able to do a mass change in real time. And from that data set, I was able to get the kinetics of the adhesion. And I was able to, of course, calculate the maximum mass adhere for each of the bacteria, and also the affinity constant of each of the strain. So that tells me if that electrostatic interaction was higher or lower.
Did you also use complementary techniques for this type of analysis for the adherence test, or did you need? Because the QCM is a time result technology, and you also mentioned that you used TEM, I think, which is more of a snapshot. Was that used for this adherence test as well, or was that more for just the uptake of the phospholipids on the membrane?
Just the uptake of the phospholipids.
Okay.
The other complementary technique was to actually test the bacteria, the adapted bacteria with milk phospholipids, to intestinal cells. And what I saw was that only 3 strains were able to increase the 2 strains, Lactobacillus casei and Lactobacillus delbrecki, were actually had a higher binding to intestinal mucosa. Remember, this is intestinal mucosa, but because I did the co-culture of CACO2 and goblet cells, they increased significantly their binding to intestinal mucosa. So I was able to, out of the 4 strains, I was able to modify and increase the probiotic status, at least from binding of those 2 strains. Lactobacillus acidophilus, I didn't see any significant change, and Lactobacillus plantarum actually decreased that adhesion to intestinal mucosa. So I saw a little bit of everything.
Yeah, how big was the improvement for the strains where you did see an increase?
Well, for Lactobacillus casei, what I had in the control was around 1.8 colony forming units.
Yeah.
Not 1.8. 1.8 times to the 6th, or 1.8 million of colony forming units per milliliter. Whereas after the treatment, I was able to increase it almost to 3. Okay. 3 times to the 6th colony forming units per milliliter. And for delbrecki, the initial binding was very low, around 0.2 times to the 6th colony forming units, and I was able to increase it to 1.2. So that was a lot. Yeah, that was really a lot.
Yeah.
So I did
Okay.
have a very significant difference. But in the end, what happens that I was able to um, make I was able to prove that milk phospholipids indeed change the chemistry or the biochemistry of those 4 strains of lactobacillus, right?
Mm.
Like I mentioned, adherence and probiotic is very strain dependent. It's
Mm.
strictly strain dependent. So I wasn't expecting the same behavior in all of the 4 strains. So I got really different results for each and every one of the strains. But I did prove that milk phospholipids change
Mm.
the addition of lactobacillus either to the gold sensor, so binding mediated by electrostatic interactions, and also they changed the addition to intestinal mucosa.
Mm. So this
So
is.
from now on, it's just the starting point of another area of research that I want to pursue,
Mm.
where I want to find the a sensor. Maybe I can design it. I don't know. I'm gonna try everything that there is. I want to find a sensor that can give me a more direct link to intestinal mucosa.
Mm. So what, I mean, I'm thinking, what are the next steps? What questions are you asking that you need answers for?
Oh, well, first of all, I want to start looking at the genes that are changed by this by adding the milk phospholipids. In our research group, we have studied that a little bit, but we have to go further. We have seen that changes that sorry genes that promote mucus binding get altered in some of the strains. But of course, we have at least in our work, we have a library of about 120 different bacteria that can be used as probiotics. So we have to do that for all of them.
Mm.
And so that's the genetic part. We also need, or I want to develop a screening system using QCM, a screening method using QCM that allows me um, to say, okay, this probiotic will increase the adherence to intestinal mucosa if I run this QCM method under these conditions. So I have to find the sensor. I have to find maybe a first layer. I've been trying to, I have prepared biosimilar mucus and try
Mm-hmm.
to bind it to a gold sensor. It didn't work. So I wanna work on that and kind of optimize that so I can have a good screening method.
Mm. And how many are those strains that you need to screen?
Well, as much as possible.
Yeah,
But
because
initially for the method, I would say about 10 different
yeah,
strains.
because you mentioned that there is a library, like World Health Organization has a library. I think you said with a number of strains that are probiotic. How many are in that library? Do you know?
Not a lot, believe it or not, because even though there are thousands of strains, not all of them are considered probiotics.
No.
I don't know exactly the number, but they're not a lot. It could, I don't know. I have no idea.
Okay. 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 Yeah. So there are some interesting next steps for you, and I mean, and like it seems like there is still a lot to learn on this in this area.
Yeah,
As
there's still a lot of things that we don't know, but at least we're starting to look at them to try to understand how the milk fat globule membrane could be something positive. Not only if we ingest it directly, but also if we feed it to probiotics, and maybe we can make better probiotics for
mm,
human health.
mm. That's fantastic. Such an interesting research area, and I'm really looking forward to what's going to happen next. Uh, and I mean, I think it's really, we've only seen the beginning of this whole, uh, like the knowledge of the microbiome and the role it plays and how we can improve it or support it and strengthen it and so on. And of course, diet is very important in this aspect.
Exactly. And through these experiments, we can highlight the importance of uh, ingesting dairy products, because we see more and more people that the trend is to stop consuming milk and dairy. However, because all the research that is being currently done, people think that we all know the components of milk, but it turns out that no, milk is such a complex food
Mm,
that every day we see more and more different molecules that are beneficial to human health, definitely. So we want to, by doing this research, we want to boost
mm,
the concept of drinking milk and dairy products. They're not bad.
mm. Okay, great. So I think we will end with that. Such a fascinating topic. So thank you for sharing your knowledge and research with us. And to our listeners, I want to say thank you for listening to this episode with me, Malin Edvardsson, and Dr. Joana Ortega-Anaya from Ohio State University. And I also would like to mention that if you're interested in surface science and related topics, you should check out our blog, the Surface Science blog. Thank you and take care.